we're going to take a look at kind of how cells work and how they're put together. Really, when we start talking about anatomy and physiology, cells are kind of a key thing in that really everything that happens is in honesty in terms of physiology, and that is really coming back to stuff at a cellular level. The basic building blocks of plants and animals, it's what we're made out of. It's the smallest unit of anything that actually has all those things occurring that are involved with life. There's homeostasis at the cellular level. So really, we're going to take a look at how we get some of these cells, how things get in or out of cells, but really the first thing here is just going to be something that honestly I think most of you have done at one point or another in different courses. Biology, for example. We're just going to be walking through really quickly some of the parts of the cell. Again, trying to put a little bit more focus on when we get to some of the systems, some of those organelles that are in cells that are going to be more key to certain cell types is some of those things that I might point out as we go through. Again, lots of different cell types in the body. I'm going to say overall they say there's about 38 trillion cells in the body, so really kind of giving you that idea of like how many cells there are that put us together. It's quite crazy, but you can kind of see here, these aren't to scale, but you can see all the different kind of morphologies of these cells based on a little bit what they're going to be doing. So really what we're going to be taking a look at is the first key question here, which is what is a cell? How does it work? So this is a composite cell. It says a representative cell, more of a composite cell. Most cells would never look like this. Again, we have extracellular fluid surrounding this cell. The fluid on the inside is that interstitial surrounds it. Intracellular fluid or cytosol is on the inside. And really what defines a cell is the cell's plasma membrane. It's what determines what's inside the cell, what determines what's outside the cell, and if we didn't have a cell membrane, inside would be outside. There is no homeostasis, and that cell is going to be dead. In fact, that's one of the ways that we kill cells off is by bursting those cells. So again, the cell membrane allows us to physically isolate the inside of the cell from the outside, and by kind of deciding what we let in, what we don't, we can really regulate the internal environment of the cell relative to the outside of the cell. What we're going to see is this membrane is what we call semipermeable, meaning some things will get in, some things will not. So it's impermeable to some things, fully permeable to others, and there's certain things that can get in depending on signals and other stuff like that. And what we can see is this is actually made up as what is called a phospholipid bilayer. Lipids do not really like water. By putting a phosphate head on them, they have something that can deal with the water that's polar. And what we're going to see is the bilayer, they kind of go tail to tail, something like that. And overall, when we look at the membrane, there's definitely more phospholipids than there is proteins, but the proteins are much larger, so by weight it's about half and half lipid and protein. You can see those phospholipids right here making on the membrane, so the phosphate head with the fatty acid tails, and they're lined up tail to tail, like I said. One of the things that we'll see with this is that they are free to move. This is what we call the fluid mosaic model. Each of these lipids can move freely past one another, with the exception of those that have some cholesterol helping to hold it together. Areas where we want this membrane to be more stable, there might be more cholesterol in it. Areas that we want to stay grouped together, you might get more cholesterol in those areas that kind of connect those tails and can create what's called a lipid raft. So this is kind of showing you lit up in green the plasma membrane. Again, the main thing that's making this up is those phospholipids, that phospholipid bilayer with the hydrophilic heads and the hydrophobic tails. There is some sugar-coated lipids called glycolipids that are going to be part of this, as well as about 20% of this is those cholesterol holding together those phospholipids that I was talking about. Again, make two sheets, line up tail to tail. One of the things he's going to do is really a lot of non-polar substances can't pass through these. Lipids can usually freely travel through, but they're going to really limit a lot of other substances from traveling through. And again, you can see the phospholipids on this image right here. And it has the tails as well as the phospholipid heads lit up on this in the different pictures, and showing you the outer and inner layers. Again, the cholesterol, like I was saying, it helps stabilize this membrane. Cells that have to be in areas of higher temperature, you're generally going to see more cholesterol. Ones that are in cold areas, you're generally going to see less cholesterol. I mean, if you're looking at cells of more extreme organisms. Again, the glycolipids help with cellular identification. It's on the outer part of the phospholipid. It kind of coats the surface of the cell, and again, helps with cellular identification, among other things. And again, we can see the phospholipid bilayer with these different proteins on the inside of it here. Those glycolipids that are making up part of that glycocalyx. We're going to see membrane proteins are found within this. Some of these can be what we call integral proteins, which means they're integrated into the membrane or fully through the membrane. Peripheral proteins, on the other hand, might be on the intracellular or extracellular surface, but do not pass completely through that membrane. And these are going to do a lot of different functions for cells. And again, depending on where we get to later on in the course, we're going to see that a lot of these we'll come back to. You'll have specialized membrane proteins that are maybe key for a particular cell type. And these you can see the membrane proteins. The one going fully through is that integral protein. The one on the surface is the peripheral. And again, integral proteins cross entirely through, which means they have to have hydrophobic regions as well as hydrophilic regions because they're interacting with those nonpolar tails that are hydrophobic, as well as interacting with the phospholipid heads, as well as water on the inside or the outside of the cell. Where the peripheral proteins, they're not traveling fully through this membrane. They're only on one side or the other. You can see the ones traveling through here. You can see now in this picture the peripheral ones. We're going to see these can do a lot of different functions, and a lot of these we will come back to. So some of these can act as transport proteins, are going to allow channels or going to be pumps that allow things to move through these cell membranes. So stuff that can't freely travel through the membrane, we might pump through or provide a channel for. There's different receptors that let us receive hormones and transmit that signal to the inside of the cell. We'll talk about that in another one of these little episodes here. Things like neurotransmitters, things like hormones from the endocrine system. There is certain things that allow our cells to identify themselves as self, or sometimes certain membrane proteins that will basically present things that are being made within the cell or things the cell is taking in that they can put on the surface and help distinguish these cells from infected cells and other things like that. There is membrane-bound enzymes, as well as anchoring proteins and cell adhesion proteins that help these cells connect to different areas or to one another. And again, this kind of shows you classified by position as well as classified by function. I'm not worried, honestly, that you know all the functions. I'm not going to ask you an essay that's going to say what are the 6 different or 7 different functional types of these proteins. That being said, we are going to learn about all of these throughout AMP1 and AMP2. So knowing what they are a little bit is going to help you out as you go along. Some of these membrane proteins that are involved in helping the cells hold together, and again, you can kind of look at us, we have 38 trillion-ish cells. Obviously, they're all stuck together. So these cells do need to be able to hold on to one another a little bit. So we do have some of these membrane proteins called things like tight junctions, also known by a much more difficult name, the zona occludens. What these do is help attach cells to one another. So fuse different parts of the plasma membrane together and hold it in place. Again, this makes certain cells very good at not letting stuff get in between them. Things like epithelial cells in the intestinal tract, cells in the bladder, so we don't want the urine traveling between the cells. Desmosomes, these are more like spot welds. They hold the cells together at a single point. You might have a ring of these around the cell. A lot of times we see these in areas where these cells might get more stress on them, so things like cells in the skin, where you can get kind of different types of stressors on that. Hemidesmosomes are kind of these half ones here that attach that cell to the underlying tissue, for example. Gap junctions, we will talk about this, especially when we get to cardiovascular and talk about the heart. But these are gap junctions. These are membrane proteins that create channels between cells, so substances from one cell can travel to the other. And this just shows you examples of each of these. So you can see this with the integral and different proteins, kind of an overall cellular membrane here. The cytoplasm is that fluid inside the cell, and it's what it's going to have all the different organelles in, and that's what we're going to talk about in the next little episode of this stuff here, is taking a look at both the non-membranous and the membranous organelles. But these different organelles are found within the cytosol or cytoplasm. The fluid is the cytosol, the cytoplasm is all the stuff together in here. Again, we have non-membranous organelles. These are ones that do not have a phospholipid bilayer making up the organelle, the cellular structure. Things like the cytoskeleton, microvilli, and the rest of this you can see here. Membranous organelles, these are going to have a phospholipid bilayer, much like the cell membrane, that is going to make up kind of the structure of these particular organelles. And we'll see things like the endoplasmic reticulum, the Golgi, peroxisomes, mitochondria as examples of those.